I'm Max, and this is Emma. We are both students at the University of Chicago in Kathy Pfister's lab. And today we're going to talk to you all about microbial exchanges in the bowl kelp-microbe cystis Leucana. So the first thing that I want to do is just introduce everyone to the biological system that we work in. So the vast majority of the Pfister Lab fieldwork happens on Tatoosh Island. This is a little pictogram of Tatoosh. Tatoosh is part of the Makah Indian Nation tribal land. And this ecosystem in general is a high research priority for the Makah tribe, especially as it relates to marine mammal ecology. And it's also really important in terms of kelp ecology, because there have been really strong, healthy populations of kelp on Tatoosh. So we're really grateful to work in this system. And both of us, I think, are really hoping that as much as possible, our research shed some light on both like causes of kelp health and maintenance of kelp health in this really pristine ecosystem. So to zoom in on the kelp, as you guys may have seen in our flyer, kelp are home to some marine mammals, including seals and sea otters. So kelp are considered foundation species for this reason. They provide food and shelter to lots of different organisms in the same environment. The part that we're going to talk about for the most part of this presentation, though, is the role that kelp have in nutrient cycling. So kelp are kind of experts in taking things from the atmosphere and releasing it into the surrounding seawater and vice versa, moving things from seawater into the atmosphere. So one of the things that is really well known about kelp is that they are good carbon fixers. So they take up a lot of CO2 and they release that into the seawater as biologically available organic carbon. They also do a lot of uptake of nitrogen compounds, and kelp are considered one of the major contributors or major sources, I guess, of organic nitrogen in the intertidal coast. So this process, which is all kind of part of like a healthy, day-to-day life of a kelp, is also really foundational for providing nutrients to other organisms and keeping those geochemical cycles going.
Yeah. So to zoom in on that a little bit more, like Max said, bulk kelp release biologically available nutrients, and they're taking up and releasing nutrients at really impressive rates. So these are huge organisms, and we can track the nutrient stoichiometry like within kelp blades and also around in the surrounding seawater to kind of have a metric for tracking the health of different kelp populations. So as we know, kelp are taking up dissolved inorganic materials like carbon species and releasing oxygen and dissolved organic carbon. And so we can calculate rates of kelp photosynthesis and have like a proxy for carbon fixation and growth by measuring these different compounds. Kelp also are involved in taking up inorganic nitrogen species. And nitrogen is often like a limiting nutrient in the seawater. So this kind of gives us another metric of the overall productivity of kelp in these different environments. And we can track also like maybe some of the less commonly studied processes like halogen metabolisms. And Max is going to talk a little bit more about this later on. But kelp are also major halogen cyclers. So they're taking up like iodine and bromide in the seawater and releasing different iodinated compounds that could play a role in like kelp immunity and pathogen defense. So these are just some of the kind of biogeochemical cycles that kelp are involved in. And really this cycling of these elements really contributes to kelp beds having a strong effect on their local environment. So previous work in the Pfister lab and other researchers have showed that looking just at the seawater within a kelp bed versus like seawater not where kelp beds are, there are stark differences. So in kelp beds, we see like higher temperatures, higher pH, oxygen, and aragonite levels. As they're taking up dissolved inorganic and depleting the dissolved inorganic material, they're increasing the amount of dissolved organic material released into the seawater. And like we said, like these iodinated compounds and higher amounts of ammonium and other nitrogen species due to higher amounts of animal activity. So we're really kind of like trying to start to understand all of these nutrients and exchanges as currencies of exchange, and being more motivated to understand the health of these foundational species. And so then I'll also just mention that the Pfister lab is noticing that on different kelp populations of like variable health, there's distinct microbial communities that are associated with these kelps. So folks in our lab, like Max and I and others, we're trying to understand the role of the microbiome living on these kelps in terms of biogeochemistry and host health. Yeah. So to set the scene into our study system, bless you, why measure all these nutrients and track the nutrients in kelp health? Well, bull kelp is in decline throughout Puget Sound. So our lab focuses on Washington State bull kelp populations that range all the way from the outer coast, where you see Tatoosh Island, and deep into Puget Sound in these inland waters. And we're seeing that kelp are in decline, and this corresponds with an environmental gradient, where on the outer coast, where you see Tatoosh and Bowman, there's a lot, it's characterized by lower temperatures and higher upwelling events, so more nutrient availability. And thus we're seeing like healthier and more numerous kelp beds. And that actually corresponds to more diverse and abundant microbial communities that live on the surfaces of these kelp. And then in contrast, as you travel inland towards Puget Sound, the seawater temperatures are higher, you have lower nutrient availability, the kelp are less abundant, and correspondingly, the microbial communities associated with these kelp are less diverse and less abundant as well.
So the real goal of understanding microbial exchanges, I think for both of our projects, is to understand how microbes are contributing to kelp phenotypes, especially those that are associated with health. So this image is a microscopy image, and it is here primarily to show you that every single color here is a different bacterial taxon. And in healthy populations of kelp, the blades are just covered in these microbes. So they are dense, they're abundant, and they're diverse. And this isn't unique to kelp. So seaweeds and a lot of other marine hosts tend to have bacteria on their surfaces at between 10 to the third and 10 to the 7 times the density in surrounding seawater. So not only are these bacteria really dense on their hosts, but we know from various examples that they're also providing some kind of functional assistance to their host in a lot of cases. So in some seaweeds, research has shown that the microbes can provide key nutrients that are critical for normal development. There's also theory suggesting that microbial communities can prevent colonization of biofouling organisms, including pathogens. So this might be important in terms of kelp health, in terms of keeping your blades clean and photosynthetic. And in a red seaweed, there was a study last year that showed that the addition of a symbiotic bacteria could rescue a disease phenotype in red algae. So the importance of microbes in contributing to overall kelp health and different kelp phenotypes is clearly important. It's likely to be a large influence on the kelp health, but the mechanisms of those interactions are still really poorly understood. So part of that is because of this taxonomic diversity and this ecological diversity of the microbes that are present. We have very different microbes with very different genotypes, and we come up with this question of who is doing what. So we know that there are certain kelp phenotypes we're interested in, but it's not clear yet whether we can, distinguish the phenotypes that are attributable to kelp versus those that are attributable to microbes. And it's not generally clear what exactly the currencies of exchange are that pass between hosts and microbes. So as a more concrete example of what we're talking about here, we know in this system that inorganic nitrogen in the form of ammonium is taken up and converted into organic nitrogen, like glycine or other amino acids. So one possible way that this could happen is the most straightforward hypothesis that kelp are actually taking up ammonium, turning it over, and incorporating it into their amino acids. But given what we know about the role of microbes in other systems and their abundance on kelp, it's also possible that the microbes are actually taking up the ammonium and they are converting it into amino acids and releasing it. And when we measure the total organism release of organic nitrogen, it appears that it's coming from the kelp. And then there's also the scenario that, we have a microbial uptake of the ammonium, but there has to be some interaction between the microbial genes and the byproduct and the kelp genes. So it could be that the turnover in this process, this entire metabolic pathway, actually depends on genes that come from both the microbe and the host. So the big problem is, how do we distinguish between these various possibilities? I'm going to offer a bit of a theoretical approach and then talk about how I'm trying to apply that in my research on iodine metabolism. But the first step in understanding microbial contributions for my project is definitely identifying what are the component parts in the system. So Bordenstein and Theis are researchers that have worked on host microbe interactions for many years. And they propose that we should think about these interactions and the contributions of microbes in the context of G by E interactions. So in this case, G stands for genotype. So we can imagine the genotype of the kelp as GK, the genotype of the microbes as GM, and some environmental variable as E. And the expectation under this model is that the kelp genotype and the microbial genotypes are both going to respond to that environmental variable. But we should also expect that they're going to be interacting with each other and potentially responding to sequence change in either component, that microbial or the kelp genotype. So we have to still answer the question of whether we can experimentally test this model of host microbe interactions. You might not be surprised that I'm going to argue that yes, we can. And iodine metabolism, I think, is a good candidate system for asking this. We have in this particular system an E and two Gs that we can look at. And I am building this model with the assumption that the environmental variable iodine is taken up based on some genetic trait in kelp. There's some conversion of iodine that will affect the genotype of the microbes, and the genes of the microbes may facilitate release of the modified product. So the data that support this kind of start with the fact that iodine is abundant in the ocean. And we know actually that it's taken up to huge concentrations in kelp, and this is facilitated by kelp-derived proteins. So that's our genotype of kelp. The actual relationship between iodine metabolism and the microbiome is really poorly understood, though. So we know that these iodinated compounds are coming off of the kelp microbe whole organism, but we don't know yet what are the effects of the iodinated compounds on microbes, or whether or not microbes affect the production of these iodinated compounds. So this kind of theoretical foundation leads to these two questions that I'm going to try to address in my thesis. First, with a growth inhibition assay. So pretty classic microbial biology here, but the idea is to take iodinated compounds that you can see kind of at the top of that little simulated 96-well plate. Apply those iodinated compounds to bacteria, some that are associated with kelp and some that are not associated with kelp, and see whether or not there are differential growth rates between those treatments. So the idea here is to see whether or not bacteria that are associated with kelp, which we would expect to have a genetic interaction with the kelp, have some different response to this environmental variable, the iodine. So these are some mock results that might suggest that kelp-associated bacteria are more tolerant of iodinated compounds than non-associates. In that case, we would expect to see perhaps the iodinated compounds are generally bad for microbes, but less bad for the kelp associates than for the non-associates. And this would tell us something about the potential relationship between the kelp and the microbes in this scenario. Maybe the kelp is using the iodinated compound for some kind of regulatory microbial relationship. Another kind of direction that we can ask the question is, what is the effect of the microbial community on the production of iodinated compounds? So rather than assuming that the iodine has a negative effect on microbial growth, perhaps it goes the other way around. To get at this question, I am this summer hopefully going to do an antibiotic treatment assay. And the basic layout for that is to take 2 blades of kelp, apply an antibiotic treatment to one, and not to the other, so that this will be a proxy for a kelp with an intact microbiome and a perturbed microbiome. And then just compare between those 2 treatments, what quantity of iodinated compounds are released. If we see that there are fewer iodinated compounds from the perturbed microbiome, we might draw the conclusion that and there's the mock data that goes with that, sorry. If we see this kind of scenario, we might assume that there is a genetic interaction between the microbial component and the kelp component. You lose the microbe genotype and you lose the iodinated phenotype. So these 2 experiments are part of the ways that my research is going to get at this question of like microbial contribution to kelp health. But this is a very targeted and limited system. We kind of know already what the compounds of interest are, what the proteins of interest are. If you remember our kind of big picture, this is a much broader problem to solve, and we need broader kinds of data to answer questions about these general cycles. So Emma's going to talk a little bit about that.
Yeah, great. So I guess how can we kind of further explore other microbial metabolisms that are associated with the bulk kelp? Because presumably there's the microbes are in really high abundance on these surfaces, so there's a lot of metabolisms going on. So some of the previous work, a previous student in the lab collected some metagenomic information. And so basically, here we're looking at some microbial genes, basically from the surface of bulk kelp, that are involved in various metabolic pathways. And so what we see is a really high diversity of metabolisms that are represented across different kelp-associated bacteria. So the top here is the different phyla that we get. And the important thing to note, I think, is that the genes aren't like uniformly distributed across taxa. Like, for example, if we look at the nitrogen cycling, there's various different taxa that contain genes to do these different functional steps in like nitrogen reduction, for example. So I think this type of data is like really informative for us to like form hypotheses and think about like what direction to go in terms of microbial metabolisms, but it's not necessarily getting at like the mechanistic, explanation of these metabolisms between kelps and their microbes. And also they're really large data sets. This is just like one subset of the data, right? So how can we kind of make sense of these huge data outputs you get from like sequencing information? And so one way that I'm proposing to kind of like analyze this data is to think about a core microbiome. And so that is defined in a lot of different ways, but basically used as a way to identify key microbes with respect to their host. So you can look at a common core, which is sometimes defined as like just taxonomic prevalence or overlapping bacteria across samples or host types. A temporal core, which would be tracking like covariating taxa over seasonal data sets or something like that. Or honing in on like a functional core, so thinking about specific microbial genes that may be important for host biological function. So in efforts to kind of like start defining and narrowing down our microbial metabolisms of interest, and given that we have such like diversity and abundance, we started to cultivate some of the organisms in our lab. So we have right now 16 microbial isolates that were cultivated from the surface of bulk kelp. And so this is, again, just showing like a subset of some of those metabolic modules or pathways that are present in the 16 different isolates that we have using whole genome sequencing. So like, for example, the vitamin Bs are known to be like synthesized by different microbes, and that the vitamins Bs could be used to like uptake by different eukaryotic hosts. So that's some sort of like microbial metabolism of interest. And hopefully this allows us to kind of like more mechanistically test our hypotheses about like certain metabolic pathways that may be important for the kelp hosts. Yeah, so then quickly just some questions that I have for my own thesis research would be how does the core microbiome affect host health? And so this could be done using like an antibiotic treatment, followed by inoculating like a germ-free kelp host with some synthetic community that represents the core microbiome, and then monitoring host health like carbon fixation or nutrient uptake rates. And also keeping in mind and maybe looking more into the gene expression of the microbes themselves to see how they may act differently when they're attached to like a healthy or an unhealthy host versus just living in seawater. And then these are just some photos of my last field season of like trying to test out this method of incubating kelp and giving them an antibiotic treatment. And another set of questions I'm interested in pursuing is being able to like track the nutrients between this kelp microbe system. So asking how kelp-associated bacteria may use these nutrients that are exuded by kelp, and vice versa, maybe the bacteria are providing some reduced form of nitrogen to the kelp host directly. And so I could do this using like a tagged nutrient assay. So it's also called stable isotope tracing, where you incubate kelp blades with labeled nitrate and then feed their exuded dissolved organic nitrogen to like a microbial community and ultimately measure the level of nitrogen in that microbial proteome. And finally, kind of the last set of questions I have is understanding how these like core microbes may compete or share resources amongst each other. So they're living in a community on this kelp blade, and I could use pairwise competition experiments to kind of understand these types of interactions, as well as cross-feeding experiments to be able to, again, track like how a nutrient is used by one microbe and then released and used by another. So that's basically all the data and whatnot that we wanted to share. And just to conclude, we've been, trying to understand these microbial symbionts that represent this like really critical but kind of understudied factor in kelp health and their life cycles. And Max and I are interested in looking at metabolites like inorganic nitrogen and halogenated compounds that kind of represent these like currencies of exchange between microbes and host. And ultimately, we're hoping that like parts of this research will inform kelp restoration efforts and different management of this really important foundational species. So yeah, thank you all. And I think that's basically all I have.
Great. So I'm excited to tell you all about some pretty new work that I've been up to looking at kelp diseases in the nursery. If you've never seen a kelp farm, that's what the slide background is. So you have all the buoys kind of floating at the top and all the biomass is underneath in the ocean. So you can't see it all. And before I kind of dive into my research, I just want to acknowledge the lands that I go on the most often for my own work. But I travel a lot, so I go over other unceded territories as well. So just wanted to acknowledge that. So why are we studying diseases at all? Well, little is known about kelp diseases, and they're predicted to become a lot more prominent in the future, both because of the intensification of aquaculture, but also because of climate change. But that's not actually enough for a topic to be important all the time. So I want to emphasize how bad seaweed diseases actually are. And there's a pretty great review from 2019 that goes over this in detail, but I just want to highlight a few key papers that they talk about in their review. So that diseases and pests in China cost about 25 to 30% of like crop death in China. So the farmers are getting 75% of the total yield they could get because of pests in this study. And similarly in Korea with pyropia. And again, kind of same story in the Philippines. So diseases and pests are really hurting seaweed farmers. And if we combine this with a 2019 FAO report that said that a little bit over 200 million USD was the total kind of global value of the seaweed industry, both selling raw seaweeds and seaweed derived products. And we assume kind of a constant 20% loss-ish globally. That means that diseases and pests are costing the global economy over $500 million USD per year, which is quite a bit. So yeah, the economy is also hurt by diseases. My work for this project was mostly done with Cascadia seaweed that's located in Stanfield, which is the green dot on the slide here, and UBC is here. So it's pretty far away. And this work was mostly possible because of Dr. Jennifer Clark, who's the chief scientist at Cascadia Seaweed. And she was super helpful, and it was really great to get to work with her and really embed myself in the production process. If you don't think about kelp farming, it might kind of be a foreign concept for what actually happens. So I want to go over that before I talk about my disease work. Kelp, as a lot of us know, but not everybody, has a microscopic and macroscopic stage. The microscopic stage starts with spores that are tiny and they swim around, and that's what you settle on your twine that you're going to outplant. And then they germinate into microscopic gametophytes, and then the gametophytes fertilize, and you get juvenile sporophytes that grow until they're about 2 centimeters big. This is still on the twine, and that's when you would outplant your kelp. And then they grow to full size out in the ocean. So my work that I'll be presenting today looks at the 3 first photos there. And what this looks like in practice in the nursery is that just seeded spools, so that would be with spores or very young gametophytes, they just kind of look white, and you just have the twine wrapped around a PVC pipe. It doesn't look like much, but after a few weeks, you start to have the spools, they kind of turn brown and fuzzy, and those are your juvenile sporophytes starting to grow. Something that struck me when I first actually got to the nursery is just how different it is from the lab experiments that we do. So the nurseries are really dense, both in terms of the space. These garbage pails are what we use to inoculate the spools with the spores. And then after 24 hours, the spools go in these aquaria. So this space is pretty tight. You have to squeeze by. And you can also just see like the amount of biomass that's crammed together. So that causes some problems, and also just the sheer scale. It's kind of hard to keep clean because everything is big. You can't use sterile technique like you would in the lab. So you're really dealing with problems of scale in industry that we don't really think about in the lab. Okay, back to the disease. And specifically, I'm going to call this the pink stuff throughout the talk. But what happens is that you get a bunch of pink that grows on the seed spools. And we saw this at all stages of the kelp. This picture was actually 24 hours after moving the spools to the aquaria. So it's, for those of you who grow kelp, it's like 48 hours after spore release, which is not very old. And we saw that the pink arrive like right before outplanting. So it doesn't seem to be associated with a particular age of the kelp, but you obviously don't want to outplant this because that would be probably not ideal, thinking about protecting the ecosystem around the farm. And also, as the pink spreads, it actually kills the kelp on the spool. So even if you wanted to outplant it, it probably wouldn't be worth your time. So my sampling scheme was as follows. We have our aquaria with the spool and then the airline to aerate the water or the kelp media. And then stochastically throughout the growing process, there would be pink splashes that appeared on the spools. So I sampled the leading edge of the pink patches, and far away from any pink, I sampled the asymptomatic region on the same spool. Then I also sampled the airline as a abiotic substrate control, and I also sampled the water in the aquaria. And the data that I got back is Illumina next generation sequencing. So I'll be talking about ASVs in the results I present. ASVs are just a unique DNA sequence that allow you to identify a bacteria. If you don't think about ASVs, it's fine. Just think one ASV equals one bacteria species, and you'll understand what the results mean. What I did is that I ran an indicator species analysis, and this lets us know if specific ASVs are associated with specific samples. So in my case, I'm interested in the ones that are associated with the symptomatic, so the pink spools. And I'll show you a plot with each sample on the x-axis, and then the size of the points is the relative abundance of the particular ASV in the sample, and the ASVs will be on the y-axis. So first, if we look at the symptomatic spools for Algicola, we see that Algicola is found in most samples and a pretty high relative abundance. So this was picked up by my analysis, and we see, yeah, it makes sense that it's enriched and highly abundant in those symptomatic spools. If we compare to the asymptomatic spools, there's a lot less Algicola in those samples. There is some, but in a lot less samples, and when it is there, it's a lot lower relative abundance than the highest relative abundance in the symptomatic spools. And if we look at the water samples, Algicola is in the water, so that means it's floating around, but it's not on the abiotic surface, which was the airline I talked about in that little diagram. So that means that Algicola isn't just randomly sticking to stuff, it's sticking specifically to the kelp, or the kelp spools anyways. And you'll notice that part of the graph is still blocked out. That's because my analysis picked up two other ASVs that are enriched in symptomatic spools. But you'll notice that both of these are a lot lower relative abundance, and they're also not consistent by year. So that tells us that Algicola is definitely the thing we should be the most interested in, and that these two may be associated, but they're definitely not the disease-causing agent. And this finding is particularly interesting because the pink stuff with my analysis is likely Algicola, but a paper from 1998 found that Algicola, sorry about the changing taxonomy, everybody, is a causative agent of red spot disease in Saccharina japonica. So most of my samples are Saccharina latissima, so it's a closely related kelp, and I found the same bacteria genus that they had as their causative agent. So that is pretty cool, I think. And what I did is that I aligned my ASV sequence with theirs, and there's 3 mismatches, which is like within that pretty short region. So I might have found a new Algicola species, which is extra cool because so far there's only one Algicola member within the genus that's described. So it might be this newly described pathogen if I can isolate it. And that brings me to kind of part B of studying Algicola. This was work done with Andrea, who is doing her honors thesis in the lab. And we got some pink spools from a completely different farm, this time on the Sunshine Coast of BC. So you can kind of see it like blobbed out in yellow, but it extends a lot further up north. And yeah, we tried to induce disease in kelp in the lab. We had healthy kelp that we grew. We tested our various assays at all different stages of life. So starting at spools and then also starting the experiments when we already had spore phytes in the lab. And what we did is that we added the pink inoculum to the kelp cultures. So both media from the pink stock, so we were keeping the stock alive, so just like the F/2 or the water that the spools were kept in, and also we snipped off part of the spool that was pink and like put that in our healthy kelp culture. And then we tried to make the disease happen by having different temperature treatments on top of just adding the inoculum, more or less water motion. We also went to the beach because UBC is by the beach. And we got seawater like from the ocean and we just added it to our cultures. We also tried filtering it to mostly remove diatoms and then adding that in case, you know, the lab culture media is different from the seawater. So maybe the seawater has something special that we were missing. But in all those cases, we were not able to get the pink to spread, which is kind of surprising because you would think disease is good at spreading. But not only that, Algicola is closely related to Pseudomonas hormonus. And for those of you who study microbes, you know that Pseudomonas is incredibly weedy. So for something to be closely related to that and not spread easily, especially when it's pathogenic, was incredibly surprising to us. And I think this is a good point to emphasize that we have little information about the scope of this problem overall. Like, do people care about Algicola? Is it a problem for growers? So I would like, if you know people who grow kelp, or you yourself grow kelp, if you could fill out this survey or email the survey to everyone you know, whether or not they have disease, to help us understand the scope of this problem. And also, if you know people who have disease who are happy to collaborate with us, we would be super happy to go swab it and figure out what's going on. So yeah, please share that. That would be incredibly helpful. And with that, I would just want to quickly plug my other thesis work. So I have a paper on bioarchive right now looking at how salinity affects the bacterial community of Saccharina, both in the lab and in the field with Connor, who is another great undergrad. And I also have other stuff that I did at Cascadia that I'm not talking about today, but I'm always happy to talk about kelp aquaculture overall. And with that, I want to say thank you to you all for listening, for inviting me to this kelp culture series, and all the people who helped me out with my research.